Hi all, in this lecture we'll see about the endospore staining technique which categorizes bacteria based on the presence or absence of the endospore. So some bacteria produces endospore during unfavorable conditions. So this endospore helps for the longer survival of the microorganisms under harsh condition, and at once when favorable condition returns, this endospore gets converted into vegetative cells and performs the normal reproductive mechanisms for the survival of the microorganisms. So these endospore are highly refractile and resistant structures. They are resistant to high temperature, heat, pressure, temperature, pH and so on. So the examples for your endospore forming bacteria are your aerobic bacteria, that is your bacillus. Under anaerobic bacteria, Clostridium forms your endospores, and these spores have a specialized acid layer, as we have already seen in case of your mycobacterium, as they have mycolic acid layer. These spores are highly resistant structures due to the presence of a specialized layer called as your dipicolinic acid layer, which is made up of calcium ions within. On seeing about the procedure for the endospore staining, smear the culture on the slide. Make a thin smear for the even distribution of the microorganism. Now stain the culture with malachite green for 1 minute, which acts as your primary stain. Heat fix it for 5 to 10 minutes so that the stain gets absorbed by the endospore. Wash it with the running tap water. After washing it, stain it with saffronin for 1 minute. Wash the slide, blot or air dry the slide, and then observe it under your microscope with oil. What exactly happens is the malachite green that acts as your primary stain gets absorbed by your endospore, and your endospore appears typically green in color, and the saffronin, which is basically pink in color, are absorbed by your vegetative cells, and as a result your vegetative cell appears pink in color and your endospore appears green in color, as shown in the image below. So we have a green color endospore and the pink or bright red color vegetative cells present. If the organism is a spore-bearing organism, then further these organisms can be classified under various groups based on the position of the endospores present inside your vegetative cells. So the first type is the organism that contains your central spore, where the spore is present in the center of a vegetative cell. Next we have your subterminal endospore, where the endospore is not present in the center as well as not in your terminal region. It is in between your terminal and your central region. Next we have your organism that produces your terminal spores, where the spores are present at the tip of your vegetative cell. Next, another type of your organism that produces your swollen sporangium, where the organism is swelled up in the region where endospore is present, and the rest of the region remains thin, but the region where the spore is present is invaginated or swollen, or it is thick in nature. Some of the examples of your endospore producing organisms can be your bacillus, amphibacillus, lactobacillus, sporosarcina, sporohalobacter. Next we have your Clostridium, Spirohalobacter, and next we have your Desulfatomaculum. So these are some of the examples of your endospore producing organism which produces varied amount of your spores. Your Clostridium botulinum produces your central spores, and your Clostridium tetanus and perfringens produces your terminal spores.